Thank you very much for joining my presentation, right? My name is Verawat from the National Center for Genetic Engineering and Biotechnology, more focusedly from the Biorefinery and Bioproduct Research Group. So our mission is on transforming bioresources to industrial application, and the topic we will talk today is from biodiversity to bioindustry. Let's say show you not in-depth research, right, but show you review what we are doing at my research group in biotech, right? Okay, so firstly, I want to introduce you, right? For our guests from abroad, maybe you're not very familiar with the concept of BCG, right? What is BCG? BCG is now the strategy for Thai economies, right? B is for bioeconomy. C is for circular economy, and G is for green economy, right? So we need to develop the technology to drive this industry, BCG economy. So what BCG economy? It can be biofuels, can be biochemicals, it can be food, it can be biopharma, it can be biomaterials, something like that. Everything on bio, on circular economy, on green technology is in the scope, right? Why Thailand invests a lot in BCG? First is that we have diverse agricultural products and byproducts that we can be used as starting material. We export a lot of sugar, a lot of cassava product, a lot of palm products, right? That can be used as the raw materials for bioconversion. We have existing industry platform, have big agro processing industry, big petrochemical industry, big plastic industry. This can be transformed to bioindustry. Okay, so what we have is we have first generation raw material, second generation, that is cellulosic material. First generation is the thing is sugar and starch. Second generation agricultural waste, sugarcane gas, cassava pulp, palm waste. Third generation they call algae, right? And then is the use of carbon dioxide. In Thailand, we produce a lot of biofuels, both bioethanol and biodiesel, available at all the gasoline stations, very commonly used. Okay, we also produce a lot of biochemicals from fermentation industry. Thailand is also the hub of bioplastic production in Asia. We are number 2 largest exporter of bioplastic to the world market, and we also produce a lot of biospecialty functional ingredients, bioactive compounds that used as ingredients and additive in food and cosmetic, and also in pharma as well. Okay, the economic value of BCG industry is now 21% of the Thai GDP, so it's quite a lot. Right? Next, okay, okay. Okay, so this is the overall scope of my research team. Okay, starting with the raw materials, first generation, second generation, bio-waste, and carbon dioxide. We have very close collaboration with TBRC, the culture collection, that is housed more than 100,000 strains of microbial strains collected in the collection and ranked, I think, now number 7th in the world. We have the robotic screening platform for screening for enzymes and bioactive compounds set up in Thailand Science Park. So once we can identify the microbes, the potential microbial candidate, so we can further enhance its performance by using synthetic biology or do hyperproducing mutant platform. Right, and then together with good bioprocess, then we have the capability on lab scale production to pre-pilot scale production up to 300 liters. Okay, so we can produce a lot of a range of prototypes products, including enzymes and biocatalysts, competent specialty biochemicals, biocontrol agents, natural products, the bioactive compound and biomass derived product as well. And work closely with different industry, agro industry, biofuels, biorefinery, food and feed, and also for green processing. Okay, more focusedly, right? The topic I will talk today will focus on 2 of the biomolecules or organisms that we will focus. First is on enzymes and biocatalyst engineering, and second is on engineered cell factories. And this is the target products for biochemicals, for enzymes, right? And this is the industrial partner and international partner that we closely work together. Okay. Okay, so first to introduce you about the cell factory platform, right? That is on the application of synthetic biology for production of biochemicals and biospecialty. I'm quite sure that the term synthetic biology is now very hot term, hot topic, maybe not only in Thailand but worldwide, right? We also uh, develop our capability on doing synthetic biology for quite a long time in biotech, for nearly 10 years already, right? And the concept is that, okay, we have very big culture collection. So we have a good platform of microorganisms that can be used as a chassis strain for further engineering, right? So we have the platform on yeast that Dr. Verawat right? And Dr. Thanapon work quite a lot, right? On fungi, right? And also on bacteria, on microalgae as well. We are going to establish the tools, synthetic biology tools, to cover all the conventional hosts and non-conventional hosts, right? Within very short term, right? But now we can engineer and do synthetic biology in Saccharomyces, in some strain of fungi, and also in some bacteria already, right? So we have good capability on doing synthetic biology quite well, right? We have the technology on machine learning to design the pieces of DNA, the promoter, the construct for building up the pathway in the chassis strain, right? Okay, give you more examples, right? Okay, so suppose this is a yeast cell, right? So what we engineer with the yeast first is on the utilization of substrates. That can use different kind of sugars, can be used glycerol, and or make it can be used cellulosic sugar, not only glucose but also other kind of pentose as well. And we can increase its tolerance to some specific conditions using either synthetic biology or by adaptive evolution. Together with use of machine learning, that will go to show you some more examples. The target product that we currently work on by the Microbial cell factory team is different kind of antioxidants, bioplastic monomers as D-lactic acid, fragrance compounds such as the aroma wood, no, agar wood aroma. In Thai we call Mai Grisana. And also some chemical precursor for organic synthesis of value-added compounds. And we have the system for so we can have the cell factories, right, for utilization of different kind of alternative carbon sources. We have the bioprocess development platform, and we are going to set up the facilities for genome scale engineering, the automated and robotic system very soon. Okay. This is examples of the beta-carotene yeast that the Microbial cell factory team has developed. Okay, you know beta-carotene, right? Beta-carotene is vitamin A precursor and have quite a big market. So usually beta-carotene is extracted from plant, but if we want to produce it in a very large amount, and if we want to specifically produce it by using microorganisms, right? We can engineer the yeast, right, to produce this beta-carotene. This is the pathway of beta-carotene and other production of other carotenoids. So Dr. Verawat, Ricky, right, not me, right? Okay, so he upregulated 8 genes and do heterogenization for another 4 genes, right? This is the chassis yeast, the common Saccharomyces. This is the one that produces lycopene. That is the pigment in tomato, so you can see red color. And this is the one that produces beta-carotene. That is the pigment, let's say, in carrot, right? So you can see it's orange color. The yield of the beta-carotene that you can produce is, let's say, is 40 times higher compared to the yield we can extract it from carrot. That's quite impressive. It's now being upscaled with the collaboration with the industrial partner. Okay. Another example is from Kun Varasirin that just sit over there. Right? Okay, this is on the engineering of D-lactic acid bacteria and further do hybridization to improve its tolerance to inhibitors in cellulosic sugars. this is the pathway on production of the lactic acid, right? Starting from glucose, right? And we engineer the pathway on production of the lactic acid. In order to increase the yield of the lactic acid, we knock out these two genes that give the size reaction for production of glycerol and also for production of ethanol, right? So we block these two genes. So you can see that the amount of lactic acid of the lactic acid bacteria is maximized for the double knockout strain. But the thing is that the growth of the strain is quite low, right? Since the product seems to be a little bit toxic, right? So the way to improve the yield is to do, let's say, conventional hybridization, right? Combine synthetic biology technology with very common, with very conventional technologies on hybridization. So we select the strains of Saccharomyces cerevisiae that has good tolerance to inhibitors in cellulosic waste, right? That is some kind of furans, some kind of organic acid, right? And do hybridization, crossing between the wild type strain with good tolerance and the lactic acid producing strain that can produce the lactic acid but has not good tolerance to the inhibitors. So we do crossing together, and you can see that the resultant strain can produce a lot of the lactic acid, right? And also with much improved tolerance to the inhibitors in the cellulosic waste, right? And this one has been published together with the team in Wageningen University in the Netherlands. Okay. Another example I want to show you for synthetic biology work is on Saccharomyces cerevisiae engineering for production of ethanol at high temperature using machine learning technology, right? Okay, so for the machine learning and AI guided synthetic biology, we have very good collaboration with researchers with professors in Shanghai Jiaotong University in China, that they are very good in yeast synthetic biology and in AI technology. So the concept is that we want to optimize the construct for production of ethanol at 40 degrees Celsius, right? Why at 40 degrees Celsius is that when operation is at higher temperature, it will save a cost for cooling water, right? Okay, so we do optimization on the expression of these three genes, that is the three genes in ethanol production pathway, right? With combination of 5 promoters with different strength. So in total, right, we will have 126 strains in the library. So we construct all these strains in the library and do checking its performance, its metabolic profile, the ethanol yield, glucose neutralization, and collect the profile for these strains at 30 degrees Celsius, right? And then we fit the data to machine learning model, 5 models, right? Different kind of machine learning algorithms. And then we predict based on the information at 30 degrees Celsius, we predict the optimized combination of promoters to this set of genes that will work well for production of ethanol at 40 degrees Celsius. This is the concept, right? This is the result. You can see more information in the paper I published already. This is the information at 30 degrees Celsius. We fit to the XGBoost model for machine learning, and then we can rank the importance of each promoter in this order using this information as the platform for machine learning at 40 degrees Celsius. So in conclusion, right, so we can get 7.4% increase for ethanol production at 40 degrees Celsius, right? 7.4% may seems not a lot, right? But in industry, it will benefit a lot, right? If it can increase from 7.4%. Okay. Finish for the synthetic biology work. We go to the second topic on enzyme platform for green industry, right? Okay. So when we talk about enzymes, right? So we means different formats of different designs of the products. The first is as the free enzymes, isolated enzymes can be in liquid, can be in powder. Second is as immobilized enzyme, we have the technology, right, on immobilization of the enzyme on solid support for increasing its stability or on for allow reusability of the biocatalyst. We have the technology for displaying the enzymes on the yeast cell surface, right, to do whole cell biocatalyst, and we can express the enzymes in the cell factory for utilization of different kind of substrate, right? Okay, this is the concept that we have. We have the high throughput robotic system, the same one, right? That can be used for enzyme engineering as well, right? And we have the technique on directed evolution and rational design on enzyme engineering, and have the know-how on doing enzyme formulation, mixing enzyme with activators, with stabilizers, something like that, right? Okay. More focusly, today we'll talk about the enzymes of production of functional sugars, right? You know, sugar industry is a big industry in Thailand, very big one, right? But the sugar industry now they face some challenge. First is that now people tend to consume less sugar, right, due to health concern. Second is that now, right, the sugar industry also produce ethanol as the products, right? But now, from the EV vehicle, right, the consumption of ethanol for vehicle will decrease significantly. No, maybe not decrease, but will not grow as fast as in the past, right? So the sugar industry, they need to do something, right, to keep their business run effectively. So production of functional sugar, not only sucrose, not only conventional sugar, if they can produce functional sugar from sucrose or from bagasse, from glucose, or from other source of raw materials, and from some sources of agricultural waste in their industry, would be very benefit for them, right? Okay, so more focusly today, I will talk about production of oligosaccharides, right, on by using enzymes, right? So what is oligosaccharides? Oligosaccharide is sugar oligomer with polymerization degree at 2 to 6. Mostly these kind of oligosaccharide, they have the prebiotic properties, right? So it can be different kind of oligosaccharides, right? From different starting materials, can be sugarcane bagasse, will produce cellulose-oligosaccharides, right? Palm waste can produce mono-oligosaccharides. Cassava pulp, they can produce cellulose-oligosaccharides. Fruit waste can produce pectic oligosaccharides, right? That for this work, Dr. Petra is sitting here, so she worked a lot on this topic, right? Okay, the first step on production of oligosaccharides is that we need to extract, right, the hemicellulose or the cellulose fraction from the starting materials. So you have the green fractionation technology using hydrothermal process or organoso process. I mean, not focus on our talk today, right? Second, we can convert the sugar, the polysaccharides into oligosaccharides using either enzymes. That's one option, and or using subcritical water, right? As another option. Today we focus on enzymes, right? We have the technology of separation and purification of the oligomer using resins, membrane, or simulated moving bed reactor, and we have good partner in the food and feed innovation center just next door to us, that they have the gut simulation system. This system, right, so can show the performance of the oligosaccharides. Okay, so oligosaccharides have different applications: prebiotic, modulating gut microbiome, anti-pathogen, immune stimulant, anti-inflammation, and antioxidants, used widely in animal feed industry, in beverage, and in food supplement. Okay, today we focus more on enzymes, right? Okay, so we have developed an oligosaccharide specific silanase, right, for production of oligosaccharides from bagasse metagenomes. Why bagasse? Why bagasse metagenome? Is that okay? The bagasse site at the sugar factory has quite high temperature, up to 50 degrees Celsius. Should be a good source for high for thermotolerant cellulolytic microorganisms and enzymes, right? So we isolated this enzyme X11 from the messenger genomes, and then we remove the carbohydrate, the binding domain out to increase its stability, and do random mutagenesis to get X11P, and do again rational design for further engineering of another two residues to get 11XP and Q, right? So it's triple mutant in total. And for the triple mutant, it has it has optimum temperature at 85 degrees Celsius, pH 6, the optimum temperature is quite high. The Km compared to the Y-type strain is decreased 4.64. The Kcat increase, right? And the Km by Kcat, that shows the catalytic efficiency of the enzyme increase up to 14 folds. And it has very good stability at 80 degrees Celsius up to 66 minutes. And if at 70 degrees Celsius can be 14 hours. That's quite impressive. And we work together with researcher, Professor in Shanghai Jiao USD to identify the thermostability determinants of the enzyme. The mechanism on increasing stability of the enzymes from the hydrogen bonding network, that's more strength, and the generation of the novel pi amide bond. Right? This work just has been accepted for publication in AMB. Right? And we apply these enzymes, right, for production of cycloochryosaccharide that Dr. Petrat has worked closely with the big sugar industry in Thailand. Right? It's now on the progress on, let's say, on the purification of the oligosaccharides. Okay, and this is the not only the cyclo-oligosaccharides, we also work on different kind of sugars, right? That you can see that many of the work can now has been upscaled at least to the pre-pilot scale already with our industrial partner. Okay, okay, we'll just have a little bit touch on the last topic to show you the examples of the product that we have developed with the industry. Some has been licensed to the industry already by our team, right? Okay. Okay, so what we have is that, okay, we have big culture collection, we have the robotic system on screening, and we have the microbial reactor platform for optimization of the bioprocess and further upscale in the bench scale reactor into the pre-pilot bioprocess bioreactor, and further in the near future to the EECI Biopolis that will have very big world-class bioprocess facilities. This is the different kind of products that we have been, let's say, licensed or is nearly licensed already. This one is the lignin composite, right? That used for 3D printing. This one is the enzyme for reprocessing of textile. This one is biocontrol agents. It is for high temperature ethanol production. This for beta carotene production and biopolymers. This one is beta glucans oligomer for pet feed. Okay. This is the examples of the enzymes license. Okay, show you. Okay, this one. And is the dual activity enzymes containing the amylase activity and pectinase activity that has the one-step desizing and scouring activity used for textile processing. It can reduce temperature, energy, chemicals, and water used in processing, while the strength of the cotton is increased. This one has been licensed to the industry already. This one go for the second generation as a cotton pro that have applied for cotton processing, right? And has the technology that demonstrated with the industrial partner already. Roxyzyme is the superoxide dismutase that has the application in cosmetic industry that has been upscaled to very big scale already with the industrial partner. Okay. And it's the example of the beta glucan that I has mentioned, right? For this beta glucan, the researcher has identified, right, some specific fungal strain, right, from the culture collection, and then study the structure of the beta glucan that fungi can produce, and has the technology on reduction of the molecular weight of that oligosaccharide to different range and show the application of the oligomer on boosting the immune, the immune system of animals, right? And this one has been licensed to the industry already and will go for further use in cosmetic and also in food industry. And this one is the biosurfactant, the mannose erythritol lipid, right? That also produced from one yeast strain. From the culture collection, this one has been upscaled together with our partner in Chulalongkorn University and also going to work with the industry under the PMUB project, the PMUC project very soon. For this one, just show you, right? Not only for the enzymes and biochemicals, we also work, our research group, right, also work a lot in biocontrol agents, right? We have licensed many biocontrol agents including bioinsecticides, bioherbicides, bioinsecticides, and also going to do biofertilizer as well. And this one has been licensed to many industry already, demonstrate, let's say, the successful cases of technology transfer from lab to industry. Right? Okay, this one I think is the last slide, right? That, okay, as I mentioned, right, one of the bottleneck, right, on transferring the technology from lab to market is on demonstration side of the technology. Although we have some pilot plan already here in Thailand, right? But maybe not very complete. So the ministry, right, has initiated this project, the Eastern Economic Corridor of Innovation, that will have, I can say, will be world-class multi-purpose bioprocess facility and also chemical processing facilities in Rayong province, and now under construction, will be open in 2 years, right? And will open for the public use very soon. Okay, so thank you very much for your attention.
So thank you, Dr. Verawat, for your engaging presentation. So we still have 2 minutes left.
Thank you so much for sharing with us a repertoire of exceptional works. So I have a question on machine learning to optimize bioethanol production. So then you show that by using machine learning, you can get 200 something combination. Did you test out all of the combination or just selected some?
For the first round, yes. Oh, yes. Actually, if you want to know more information, I mean, talk to Dr. Ricky. It's still here. Okay, no. Okay, but anyway, this work has been published already, right? And for that work, we do quite a lot, right? On constructing the strain and do characterization of the strain. I can say this is the first work of our team on application of AI on machine learning for synthetic biology, right? And now this technology platform is quite established. We are to apply for different yeast, for the yeast producing other products as well, right?